So thanks to everyone for joining our fourth, I think it is, pilot session from our In-Silico Airlock. My name is Professor Alex Frangi. I'm professor at the University of Manchester, and I would like to welcome you to the UK Centre of Excellence for In-Silico Regulatory Science and Innovation training webinars, where we are proudly supported by Innovate UK, the Medical Research Council, and is sponsored by the Medicines and Healthcare Products Regulatory Agency, or MHRA. As the UK's first centre of its kind, we pioneer the use of in-silico methods to enhance regulatory science and innovation, and our webinars will delve into the latest advancements, best practices, and applications of in-silico approaches. And our goal is to foster collaboration and knowledge sharing among various stakeholders to drive progress in healthcare and medical research. And we hope that you will enjoy the session and also join us to explore the future of regulatory science, not only in this, but in some of the other events. If you have missed any other previous events, they're all recorded in our YouTube channel. And later during the session, we will put the links in the chat so you can subscribe to those or follow us up on those. So today, without further ado, I will hand over to my colleague Aresu Sakeri and to David Flynn, who together with me and frankly to a large extent have them driven the development of this webinar, and who will provide the agenda and some of the next steps. And we will do probably an hour, a bit more of an hour of presentations of various people that will show different perspectives of this use case. And then at the end, we will have at least 40 minutes for Q&As to delve into any aspects that you deem appropriate. And as I always try to impress, this is important not only to have questions about the technical aspects, but I'm delighted to see that there is also colleagues from regulatory aspects, from social sciences. I noticed some of my colleagues, philosophers, as well, that have joined the webinar, so it will be really useful, health economists, to put questions if you feel that can be helpful. Today, our seminar is, our pilot is particularly interesting because you could argue that this is a pilot related to in-silico technologies or to AI and synthetic data. Is actually at the interface and shows a concrete example where the field of synthetic data can be useful and to enable certain types of in-silico trials that otherwise wouldn't be possible. So thank you with that, and Aresu and David, I'll hand over to you.
Yeah, thank you, Alex. So I start sharing my screen. Can you see my screen?
Yes.
Hi, everyone. My name is Aresu Sakeri. I'm a lecturer in medical image computing and AI from University of Manchester. Thank you, everyone, for joining today's pilot on establishing credibility recommendations for generative virtual populations, a case study in TAVI device regulation. Before I start, I would like to thank all the collaborators who have contributed to the discussions over the past couple of months. This work reflects a collective efforts of our team from industry, regulatory bodies, and academia. I'll begin by providing some background and introducing the pilot scenario, including question of interest and context of use. Then Steve will share industry perspective on the topic, and Gavin will provide insight from regulatory point of view. Then I will shortly cover model risk assessment, and then Horan will walk us through verification and validation activities for generative models. After that, Shakun will present discussions on uncertainty quantification for the generative models. And finally, we will wrap up with some key takeaways from the group, and we'll open the floor for a Q&A session. And finally, Alex will outline next steps and formally close the panel. Aortic valve stenosis and TAVI treatment. The aortic valve normally has 3 leaflets. However, in a rare congenital condition known as bicuspid aortic valve, the valve has 2 leaflets instead of 3. And this anatomical variation significantly alters blood flow patterns around the valve, and this can lead damage the leaflet and trigger inflammation. Over time, the healing process results in calcified scarring, making the valve more rigid, and eventually leading to a condition called aortic valve stenosis or narrowed valve, and this limits the blood flow to the aorta and the rest of the body. While aortic valve stenosis typically affects older individuals with tricuspid valve, often in their 70s or 80s, this happens to bicuspid patients much earlier in their 50s or 60s. In addition to early onset stenosis, bicuspid valve patients are also at higher risk of aortic regurgitation, where the valves become leaky. And this occurs when the leaflets become floppy due to weak connective tissues. Another important concern is the association between BAV and aortic dilation or aneurysm, which can further complicate the condition. In most cases with aortic stenosis, valve replacement is recommended treatment. In recent years, transcatheter aortic valve implantation, or TAVI, has emerged as less invasive alternative to open heart surgery and offering new treatment options for selected patients. Randomized clinical trials assessing the safety and performance of TAVI devices initially focused on high-risk surgical patients and later expanded to include intermediate and low-risk patients. However, bicuspid patients were traditionally excluded from all these trials due to their anatomical complexities. Notion 2 is currently the only randomized clinical trial that includes both tricuspid and bicuspid patients and treated with self-expandable devices. This trial has enrolled 138 TAV or tricuspid patients and only 49 bicuspid patients. 1-year outcomes from Notion 2 confirm that TAVI is feasible for bicuspid patients. However, the occurrence rate of post-TAVI complications is higher in bicuspid patients compared to tricuspid patients. And this suggests that clinicians should be more selective with TAVI in bicuspid patients. Despite these findings, several important questions remain, particularly regarding which anatomical features are more suitable for TAVI, or conversely, which BAV patient should not be treated by TAVI and better to be treated by surgery. Or other questions about the durability of the device or the type of the valve based on the anatomy. And these questions cannot be easily answered through Notion 2 clinical trial or similar clinical trials because of the limited sample size and also because highly selected patients. So now, having dedicated randomized clinical trials for BAV patients is perfect and ideal, but it is very challenging. That I will discuss shortly. But the level of knowledge we have for bicuspid patients compared to the tricuspid patients is not the same. And who is a good candidate to be included in these trials? What anatomical features should be excluded? And these uncertainties have motivated the design of a recent prospective single lab registry called BiVolute X for BAV patients. This registry started in 2018 and so far has enrolled only 150 BAV patients. As I said, running clinical trials for BAV patients is very challenging. It is very expensive as it is a long-term follow-up. Recruiting this underrepresented population, which represents 1 to 2% of the general population, is time-consuming. In addition, bias may occur in the enrolled patients, and depending on the trial location, participant diversity is likely to be limited. This challenge highlights the need for alternative approaches such as generative virtual populations of BAV patients using generative models and running in silico trials to provide in silico evidence to answer some of these questions. And this brings us to the hypothetical example introduced in our pilot, which focus on the credibility of these generative models. A key requirement if in silico evidence is to support regulatory and clinical decision making. Workflow of an in silico clinical trial or ICT for TAVI devices depends on multiple submodels, and it's important to recognize that each submodel credibility matters, and if one submodel flawed, it can compromise the entire trial. Generative models, which are responsible for creating these virtual populations, can be considered as a computational submodel. And in the risk-informed credibility assessment framework, each submodel's credibility evidence must be proportional to its impact on the final regulatory or clinical decision. But what is a virtual population, virtual patient in the context of in silico trial? It's not just a geometry, it's a composite digital representation driven by data and physiological realism. We start with real-world patient data and define conditioning variables for our generative model. This can include structured data like demographics, such as age, sex, weight, height, etc., or medical conditions such as hypertension or diabetes, anatomical measurements such as calcium volume, or potentially lifestyle and lab measurements. This can also include free text clinical summary as shown here. Once the generative model is trained on the real population, it allows us to sample from the land distributions to generate a sample anatomy. Depending on the structure of the generative model and the data used for training, it can also train to generate boundary conditions or material properties. But it's important to note that the virtual patient must be fully plausible given the input data and grounded in physiological reality, and should be able to support downstream simulations for device deployment and evaluation. This is a typical approach for generating virtual patient population for in silico clinical trials. We can use real-world populations such as UK Biobank or other larger scale and public data sets to train the generative model. The goal is to generate virtual populations that reflect real anatomical and physiological variability. Once trained, the generative model once the generative model is trained, we can sample as much as we want and create a very large population of synthetic patients. These synthetic cases are then filtered using inclusion-exclusion criteria from existing clinical trials to ensure the virtual population mimics real trial cohort and is clinically relevant. The core of the generative framework could be a multi-channel variational autoencoder shown in the bottom panel, and it uses multiple encoders and decoders to handle different anatomical parts of the whole anatomy, learning a shared latent space that captures the underlying variability. This shared space allows us to generate anatomically plausible 3D heart shapes, condition, and desired characteristics. And then to validate the virtual population, we compare the key anatomical and physiological features, some important quantities of interest, for example, annulus size or annulus area, left ventricle mass and volume, between real and generated population. And these histograms are statistical, and the statistical analysis on them show close agreement supporting the fidelity of the virtual population. So given this background, this pilot is to establish a credibility recommendation for generative AI models. And here we present hypothetical examples of a target population for the TAVI device in ISCT to discuss the credibility framework. But we don't execute the credibility plan. An approach is we first define in silico clinical trial question of interest. We define the model context of use, and we perform a model risk assessment. We finally provide recommendation for verification, validation, and uncertainty quantification activities required to provide credibility evidence for the generative AI models in ISCT. So the pilot scenario and in silico question of interest is that could we use in silico evidence to impact patient selection in younger bicuspid clinical trials? And the question of interest from regulatory point of view for the device is safety and performance of self-expanding TAVI devices in younger low-risk bicuspid patients equivalent to that in tricuspid patients with comparable anatomical characteristics such as similar angulation and MASH calcification grade, for whom safety and efficacy of the device have already been established in clinical trials. To answer this question of interest, as I just mentioned, in an in silico trials, there should be several submodels, but the context of use here for creating just virtual populations is that a conditional geometry generative model will be used to generate a virtual population of younger low-risk BAV patients with some key anatomical features known to impact TAVI outcomes. And that is horizontal aorta, aortic angulation greater than 40 degrees, and various grades of valve calcification. The outputs of this geometry generative model, which is anatomies related to TAVI, such as aorta, aortic root, left ventricle outflow, and calcification, are used as inputs to the computational simulation for TAVI device deployment. So we should emphasize that this pilot assumes computational models and simulations that will be ultimately used for answer the question of interest about safety and performance of the device are already validated, and their credibility assessment is not the focus of this pilot. And also other generative models that are used for generating boundary conditions or material properties or other submodels that will not, their credibility assessment will need to be evaluated separately. And we need to also define scope of the context of use. So here we assume that for this hypothetical example, just we generate, for example, synthetic anatomies. So dynamics of the heart will not be modeled. Or we can also should scope the context of use, and that we are not modeling material properties or non-geometric descriptors here. So I pass over to Steve to present insights from industry on the context of use.
All right, thanks, Arzu. Yeah, so a big picture here as we think about the capabilities of generative models to work in this workflow and really thinking about the creation of models that flow into simulation from a physics-based perspective or some other downstream activity. I think this particular application is particularly useful for industry. And there's some notes on here, big picture. What we're looking at and the benefit of this generative approach is really overcoming some of the limitations that surround current methods, statistical shape modeling, digital twinning with specific patient-specific clinical imaging data, et cetera. Those are great methods. They have important applications, but they also have important limitations. And I think this generative approach that we're discussing here today offers many opportunities to overcome some of those limitations, some of which are listed in the sub-bullets in the middle of the slide here. If you're going to need to do some filtering and you need to think about different aspects of the virtual patients you're creating, it's going to be valuable to be able to, relatively speaking, of course, rapidly and cheaply generate the inputs that you're going to need for your model. There's concerns, of course, about existing clinical populations being limited, particular applications here being rare conditions or orphan conditions, where you might not have a huge clinical database to grow off of. So there's obviously validation questions there, but I think there's opportunities from a generative perspective to fill in the gaps, if you will, with those existing clinical populations. And then this other point with respect to multimodal data being particularly valuable here. And, Arzu mentioned the fact and the point that these bicuspid patients, just for this particular scope, are younger patients, right? So when we're thinking about younger patients and we're thinking about devices that are going to be used for these younger patients, it's going to be really important for us to think about what the loading conditions are going to be, what the sort of future history, if you will, of the patients are going to be. And, as Arzu mentioned, the scope of this is really just anatomy. But I think the applications here and the thought processes and the frameworks that have been developed through this pilot can easily apply to other generative models that can fill in those gaps, that can really provide some of that longitudinal projection in terms of how these patients might behave and the different activities they're going to have. So I think it's really important to be thinking about the credibility assessment for generative approaches. And this in particular, I think, is a really attractive use case because I think it's a very practical application. But as just mentioned, these generative approaches really can be applied in a number of different ways. And so thinking through the credibility assessment aspects of generative approaches in this very concrete example where we have comparators like imaging data, super duper useful. So I really applaud the group here for the work that they did. You guys are going to see some really cool stuff as they lay out what this credibility assessment framework looks like.
Thank you, Steve. And I pass over to Gavin to present regulatory perspective on context of use.
Thank you. Unfortunately, Rene, who's done most of the sort of regulatory work on this particular pilot because his background is cardiac, can't be here today. But I think for us, sitting in the regulatory space, what we're really talking about here is we expect these In-Silico and generative AI models in In-Silico trials to have the same standards of credibility, traceability that we would in regular clinical trials. But I think the point for us is it's really important to mention that all regular clinical trials have some issues. There's no such thing as a perfect trial. Patient selection is a big deal. Patients disappearing, dropout rates, transparency of documentation, and validation of all the results. Both of us have worked for notified bodies for many years, and we see these weaknesses. And one of the potential advantages here in this model is that the risk, if you like, can be estimated, and it depends, on as Rene has said here, in the noise of the input for the model, and that's something you typically don't see often in clinical trials. There's no real attempt to estimate at the beginning how credible you might find the results. So there are distinct advantages here, and I think what we would like, as assessors rather than regulators, really, in our role, is we'd like to see the regulators really establish some more concrete and actionable rules for the assessment of in-silico evidence. And we have seen the European Commission mention in-silico in the context of orphan devices. And I think looking at this pilot, this is a relatively rare indication. You can imagine that as you scale down to children, for example, the numbers become very small. And this is a real, cause of concern that devices are not being used and innovated for children and patients with rare diseases. And it's easy to see how some of this in-silico evidence could support that regulatory decision making. And I think Rene's point here is, we accept the noise of other trials. We accept that there's no perfect trial, and we should be willing, I think, as regulators and as clinicians as well, to accept that there will be things that we don't know and that things will develop in an iterative process. And I think Rene's final point really is is looking towards maybe more acceptance with the Commission as part of the expert panel group. And for those of you who don't know, the expert panels have been set up as part of the medical device regulations to provide a group of clinicians to look at the clinical evidence that's submitted for high-risk devices. And I think if we can demonstrate that this sort of evidence is really useful and that it helps both patients, clinicians, and regulators to support the decision making, I think really that's the goal of all of these pilots. And I think this one in particular does show some, real clear links between, theoretical concepts, practical concepts, and what actually happens to patients. And I think that for us is the interesting takeaway. Thank you.
Thank you, Gavin. Okay. So the third step in the risk-informed credibility assessment framework after defining question of interest and context of use is model risk assessment. Aligned with available guidelines on credibility assessments like credibility assessment for computational models and simulations such as ASME-V and V40 for which is specifically for computational models and simulation used in medical devices. Here, the model risk can be defined for generative models, the possibility that the generative models may lead to an incorrect decision that would lead to an adverse outcome. And we assume here that the generative model is the only source of incorrect decision in in-silico trial. And so any potential harm to patients from incorrect decision in ISCT, such as those caused by the issues in the synthetic data, such as drift between real and synthetic data, and any other issues in the synthetic data should be considered here. And model risks include model influence and decision consequence. Model influence is the contribution of the generative model relative to other contributing evidence in addressing the question of interest. And decision consequence is a function of severity of the harm and the probability of occurrence of the harm, and indicate the significance of the harm resulting from an incorrect decision concerning the question of interest. So to do model risk assessment for generative models in silico trials, we have to complete these 5 steps. First, we need to identify which harms should be considered. And this is considered in the in silico trials. And this is based on the framework proposed by Favre and Bishop after answering these questions, that whether is risk medium or high, is harm impacted by device, or can harm be simulated? And if all the answers to these questions is yes, then this harm can be considered in the in silico trials. So after that, we determine the decision consequence based on harm severity and probability of its occurrence. We then evaluate scope and coverage factors, which indicate model influence, and then we estimate the model risk based on model influence and decision consequence based on the metrics I showed in the previous slide. So for this pilot, to evaluate the scope and coverage for a hypothetical example of these bicuspid aortic valve patients with different aortic angulations, the scope indicates the amount of alignment between in silico data with the clinical data. So from clinical data from a registry, we know that the range of aortic valve angulation is something between 18 to 90 degrees. And the scope considers whether the synthetic data aligned well with the clinical data, or it actually augment patient characteristics beyond the scope of clinical data, and whether the worst case outcomes or worst case clinical scenario are considered in the clinical data or not. So for our case, worst case clinical scenario are aortic angulation greater than 70 degrees. And based on the clinical data available from this registry, we could say, okay, this range is covered through clinical data, so we can consider that a scope factor of medium for this problem. And coverage, on the other hand, shows the magnitude of voids in clinical data. And again, it depends on the clinical data we have in hand during the execution plan. And by quantitatively checking the magnitude of voids or those scenarios that will be considered exclusively within In-Silico data, we can determine what is the coverage of the model. And then, for example, here we can assume that, okay, configuration addressed solely by the ISCT are expected to represent a small number of clinical cases, so a low model influence can be considered. So with a medium scope and a low coverage, model influence is estimated to be low medium. And then for a specific harm, like new permanent pacemaker, which is a common post-TAVI complication, from a real clinical data, the occurrence rate is reported 15.1%, so it is a probable complication. And the severity of the harm, which defines as an impact on patients, is critical because it's a life-threatening harm. So based on critical severity and probable occurrence rate, we can say the decision consequence is medium-high. With the model influence based on a scope and coverage of low-medium and decision consequence of medium-high, we can conclude that the model risk is medium. Now the model risk is estimated, and based on that, we can establish a credibility assessment plan and different gradation for verification, validation, and uncertainty quantification. I pass over to Horan to present these parts.
Yes, thanks, Arzu. Let me share my screen. Can you stop sharing, Arzu, so that I
sure.
can share it?
Okay, I think you can.
You can just follow your current thing.
I can. Okay, yeah.
Yeah. Cool.
Wait.
Let me share my screen. Can you see my screen?
Yes.
Okay, cool. So basically, I will briefly introduce the verification part and also the validation part. So, oh, sorry. So basically, for the verification, the verification of activity between the computational models and the generative model actually are a bit different. So the computational model is a physical driven and also the mechanistic prediction, but usually the generative model is data driven and it is based on the statistics and perform the probabilistic prediction. So if you see the table that we have performing some investigation on the credibility factors between the ASME V40 for the computational simulation model and also the generative model, you can see the most differences comes from the code verification. So in the generative model, we need to verify the whole implementation of the code and also perform the additional statistical verification. So in the calculation verification, the only differences in the numerical solver error in the ASME V40, where in the generative model, we actually check the model architecture and the optimization errors during the model training, but both of them actually sharing a kind of similar concept. So let's go through the verification activities of the code verification. So for the implementation verification, this can be roughly divided into the software or programming environment, code correctness, and the version control, model reproducibility, and API or interface. So let's using the software and the programming environment as kind of example. So one of the activity in this aspect can be checking the documentation of the library version for the programming and also identify the potential conflict of each third-party library. So the code correctness can also be whether your code implements the model architecture and also the training sampling procedure and also the metrics correctly. So for the numerical statistical verification, we consider include the numerical precision, statistical computation, and also the algorithm convergency. So for instance, the statistical computation verification activity can involve the checking the sampling correctness of the generative model. So based on this, we actually can create kind of a checklist to obtain the credibility evidence of the generative model on the aspect of the code verification. And also for each sub-item, you can also create the gradation of its credibility level. So then we can, let's say, create and suggest a risk-informed gradation of a verification activity based on those, let's say, introduce the checking list. So the credibility of the verification activity can be graded from the low to high based on the checking item. For instance, verification with low credibility factor can be there are very limited or no verification is defined or documented. Input and outputs are not verified. No discretization analysis is investigated. Well, for the high credibility, all items in the implementation and the numerical statistical verification activity have been fully verified and documented. And also the calculation error has been fully investigated to ensure each item reaches the highest level. But remember, this is actually kind of, let's say, a suggested general, let's say, risk-informed gradation of verification activity. Let's say, this gradation of verification activity can also be performed in each sub-items of those, let's say, verification aspects. So let's move to the validation activity for the conditional generative model. So similarly, let's say, considering the differences between the generative model and also the computational simulation model, we actually found it is quite challenging that we directly align the virtual population validation to the ASME V and V40. So in this scenario, we suggest to, let's say, follow another document from the FDA named Scorecard, which actually include quite a lot, comprehensive synthetic data validation activity. So the scorecard for the synthetic medical data evaluation and reporting can be divided into three different parts. So the first part is focusing on the data itself. So it's namely the data quality. So they summarize this, let's say, 7 different aspects that they call the 7 C's. So let's say the 7 C contains the congruence, coverage, constraint, completeness, compliance, comprehensive, and consistency. So we will introduce the detail of those 7 C's later. So the task specific typically is actually to validate the utility of your virtual population model. So the task can be a predictive task or a simulation-driven task that's designed for the in-silico trials. So another one is called the human-dependent, let's say, evaluation. So this is actually not task-specific, and also it is very, sometimes it's infeasible for performing the human-dependent to a large-scale data set because of the resources and the timing of the human expert. So let's go through some details of the 7 C's. So the first C is called the congruence, which is actually the fidelity of the virtual population. So the congruence is actually to assesses the degree to which the distribution of synthetic data aligns with the distribution of the patient data. So if you can see this table, so the table summarizes the potential aspects and also the evaluation metrics from the literatures that can be used to assess the virtual population in our pilot scenarios. So the aspect can be divided into the geometries, which contains the alpha, LVOT, and VOV, and the classification. And also the parameter of interest contains all of the list of the, let's say, the anatomical features that we are interested in. For instance, like the LT angulation, classification volume, LT analysis size. So the right one is a kind of, let's say, an example of the congruence evaluation in a literature that's working on the TAVI virtual patient generation. So it shows a kind of scatter plot of all the combinations of anatomical features for both the real and the synthetic geometries. So the red dot is the the real geometries, and the black dot is the real geometries, and the red dot is the synthetic geometry. So based on this, you actually can observe the overlap between the real cohort and also the generated virtual cohort in terms of different anatomical features. Also on the diagonal, the distributions of the individual features are shown in the histogram. So let's move to the coverage, which is actually the diversity and the novelty. So this actually to evaluate the extent to which synthetic data captures that diversity and the novelty inherently in patient data while aligning with the true distribution. Similarly, we also have kind of table that involve the potential evaluation metrics for assessing those, let's say, the coverage. So here is actually another table from that literature in the previous slide. So this table reports the mean and the standard deviation of the synthetic geometries, real geometries, and also those from the literatures in terms of those, let's say, those anatomical features. Let's focus on the standard deviation. Because standard deviation actually describe kind of the diversity of the data set. So you can see, for instance, for the LVOT diameters, the synthetic geometries can achieve a 3.3 standard deviation. Compared to the real geometry, it's actually enlarged the let's say the diversity of the cohort, and also is very close to the literature's the standard deviation reported in the literature. Okay, so let's move to the next two. So the constraint is defined as the extent to which synthetic data respects known constraints, which may include the anatomical, geometric, or clinical constraints. So it's quite similar to the inclusion-exclusion criteria we use in the clinical trials. So basically, you actually to check the availability of your generated data set within this constraint. For instance, in the demographic data, since we are trying to assessing the younger patient, this constraint can be the age is lower than 75 years old. So another is like the completeness. Completeness is defined as the extent to which generated data constrains all necessary details relevant to the task. So the virtual population, as introduced by Arizu, that the virtual population cannot just an autonomy, but you should also involve more information, like the age, gender, or medical condition, etc., something like this. So now let's move to the another three aspects they call the compliance, comprehension, and consistency. They are related to the privacy, transparency, and the fairness of the generated data respectively. So the compliance is more related to the ethical and the privacy concern. Sometimes it's related to like it needs to ensure that your generated data is not as the same as your training data. It should be an ensemble of your training distribution, but not a copy of your training distribution. So this is actually to prevent your generated model to make some risk of the data leakage. And the comprehension is actually referred to the how well is your documentation for the whole model and your generated synthetic data. So it's like you need to concern the model formulation, model optimization, model evaluation, and also your data and code, whether your training data has some bias in your group, and also how you generate, what's the whole generation process of the synthetic data. So basically, all of this should be documented well. The consistency is actually another very important, let's say, aspect that works to validate systematically. So the consistency, it requires you that you are not just focusing on the validation on the full cohort, but also pay attention to those, underrepresented cohort, those subgroup in your cohort, whether your synthetic virtual population can achieve comparable fidelity to those cohorts in the real world. So an example in here, you can see they divided the LVOT shape in the coverage and the diverging. So they calculate what's the occurrence in the unfiltered virtual cohort. It's like there are 62% of virtual patient is occurred in the coverage, and 38% is in the diverging of the LVOT shape. So now let's go through some, let's say, based on those 7 C's and the scorecard, you can also create a kind of, let's say, checklist. So here is actually the example of the scorecard checklist on 2 recent literatures that are working on the virtual population generation for TAVI In-Silico trials. So you can see the table, they actually perform different aspect evaluation on the synthetic virtual population. You will also see, let's say, the evaluation actually has different representation formats. For instance, in this Congress evaluation of the first literature, they actually only visualize the box plot of the 2 anatomical features. So although they perform, let's say, the evaluation of the virtual population on different aspects, you actually can grade the credibility of their general evaluation, because neither of them has investigated the credibility factor of their evaluation method for each aspect. For instance, when you're trying to, let's compare this box plot to the previous scatter plot in the previous slide. This box plot deliver a very less message compared to that, scatter plot. Which means it actually have a low credibility compared with that scatter plot, because the scatter plot not only observes the overlap between the real and virtual population in terms of the distribution of the anatomical feature, but also it can provide some insight of the correlation between the anatomical feature that are whether being really captured in the virtual population. So the whole thing actually suggests that we also need a credibility gradation of the detailed subatom as well, which is actually quite rare in the current generative model literatures. So in general, we can also suggest risk-informed gradation for the validation activities of the generative model, that we start from the data quality evaluation with the 7 C's to the utility of the downstream task, the task-dependent evaluation, and we can involve the expert-dependent evaluation. So you can still, let's say, grade this from the low to the high. So basically, the low we can only focus on the 2 important C's, the congruence and the coverage, which is let's say the fidelity and the diversity. So a very simple example method is to comparing the mean and the standard deviation of the cardiac parameters, for example, the altitude angulation between the real and the synthetic population. So then the medium low can be, we check all of the 7 C's to validate the synthetic virtual population. And then we will involve plus the task-specific evaluation to assessing the utility of the generated virtual population for the computational simulation. Then in the highest credibility level, so we can adding the expert-dependent evaluation to, let's say, not to evaluate, let's say, to manually checking. Sorry. Sorry. Yeah. Let's move on. So basically, do the manually checking on the let's say at least a proportion of the synthetic virtual population. So the desired accuracy to distinguishing the real and the synthetic data by human expert should close to 50%. So that's pretty much everything about the verification and the validation. So on the other hand, the uncertainty quantification is also a very important aspect to assess the credibility of the generative model. So I will move this to Xiaokun. Yeah.
Yeah, I have submitted a request to share the screen.
Yeah, I have stopped.
Okay, so could you see the slide? Yeah, okay, I'll make it. Maybe in full screen. Yeah, so could you see the slide?
Yes.
Okay, so I will introduce the part of uncertainty quantification. So at first, I will give a brief introduction of the related credibility assessment. So there is some related introduction in the documentation of ASME, like the credibility assessment can be divided into two parts. The first one is sensitivity analysis, and it is an important credibility factor which can examine the degree of which the output are sensitivity to the input for the genetic models. And for the second part, it is called the uncertainty quantification, which is aiming at examining the degree to which unknown or assumed uncertainties in the model inputs and which can be propagated to uncertainties in the model outputs. And actually, both analyses are very important and they are actually complementary to each other. And so what we would like to do is to create a loop between these two analyses and provide valuable insights for the genetic models. And we will introduce this loop in the following parts. And so I will give the introduction of sensitivity analysis and uncertainty quantifications and one by one. And the first I will introduce give an example of what is sensitivity analysis. Like we have a condition genetic models for virtual population. What we would like to do is to conduct a ablation study and to evaluate which input for our models can contribute meaningfully to the output. And we would like to run our genetic models with input which we can select and without this input. And like we can see whether the related variation will disappear in the output of our genetic models. Like if we select the age or sex demographics, and we can see whether the age or sex dependent variations will disappear in our model output. And there are three targets for our credibility assessment. Like for the high target, we would like to do a comprehensive sensitivity analysis. And for the medium target, we will perform the sensitivity analysis, but we wouldn't estimate the errors. And for the low targets, we wouldn't do the sensitivity analysis. And then I will introduce what is uncertainty quantification. In general, it can be divided into two types of uncertainty. The first type is called statistical uncertainty or aleatoric uncertainty. It is due to the inherent noise or randomness in the input data. So actually, it cannot be reduced by ourselves. And for the second part, second type of uncertainty, it is systematic uncertainty or epistemic uncertainty. It is due to the lack of knowledge or limited data. And actually, it can be reduced with the optimization of our models or with more information of the input data. And here I give a pipeline of the genetic models. We can see that in every process of generation, there are several types of uncertainty which can be identified. Like in the input process, there are some input uncertainties, like in the process of segmentation or pre-processing or measurement uncertainty of inputs. And in the process of generating, there are some uncertainty due to the model form, parameters, loss functions, or so on. And this can be optimized by our training process. And in the output part, there are uncertainty examples of measurements, like maybe there are limited number of measurements or statistical sampling errors and so on. And so here is a pipeline of uncertainty quantification process during the generation. And then we can give a workflow for the uncertainty quantification in our conditional genetic models. And here there are three important steps. The first step is we need to characterize uncertainties for our genetic models. We would like to identify all sources of uncertainty in input and model structure, and we can define a probabilistic distribution based on the input clinical data a priori. And then second step is we would like to propagate these types of uncertainties during the generation process. Maybe we would like to sample from the input into distributions and run our models on these sampled inputs and to generate a probabilistic distribution of outputs. And the final part is we would like to analyze the results from the outputs of training models, and like we can analyze the statistical properties, and we can evaluate whether the uncertainty levels can be acceptable for our application in virtual population. And here, I must say that if we would like to ensure our generative model can produce trustworthy outputs for our application. And we need to build such an iterative loop of uncertainty quantification and sensitivity analysis. And there are 4 steps here. The first step is the initial uncertainty quantification, where we need to define the input uncertainties and to propagate these uncertainties through our models. And this can help us to examine the output variability due to the input uncertainties. And then we would like to do the uncertainty analysis to identify which inputs are which inputs can most significantly influence of the outputs. And then we would like to do the refinement based on our previous sensitivity analysis. Like if we can find an input is highly sensitivity and highly uncertainty to our outputs, we can collect more data for that input, or we can narrow its assumed uncertainty range or constrain our generative models to reduce the variability in that region. And then the final step is we would like to repeat this loop with updated assumptions, and this can help us mitigate the most important credibility risks. And what is the most important during this process is the uncertainty traceability. And what we would like to do is we would like to trace the uncertainty of our generative models from the source. And this can be used, and this is very important for the simulation task or downstream task. And the traceability is actually a term which means that the uncertainty sources in the generative stage are explicit and traceable in our downstream tasks. And so actually what we would like our models do is that our built generative models should produce the anatomies with documented and traceable uncertainties, which can be evaluated in our downstream task. And this is very important. And so in general, there are 3 levels of risk-informed uncertainty quantification. And so for the low level, we wouldn't conduct uncertainty quantification. And for the medium level, we would do a basic characterization of uncertainties. And for the high level, we will do a comprehensive uncertainty quantification. And so next is conclusion. I will move the part to Arizu.
Thanks, Shakun. So I conclude the discussions with the importance of developing new and practical generative AI specific regulatory guidance tailored to models used in silico trials. And you can see here a list of selected list of international guidelines for trustworthy AI in medicine from this. Could you put it on slideshow, Shakun, please? Shakun.
Yeah, okay. Sorry,
I was using your slides.
I misclicked. Yeah, sorry.
Yeah, okay, thank you. So you can see a selected list of previous slide, please.
Yeah, okay.
Yeah. You can see this is a selected list of international guidelines for trustworthy AI in medicine from this publication. And some of them are for generative models, but most the issue with most current Gen AI guidelines is that they lack depth in practical implementations in clinical settings, and this highlights the need to do more work in this area and proposing more practical guidelines, particularly for generative model used in silico trials. Next slide, please. Yeah, and here I have listed some lessons learned. Some lessons that we learned during our discussions with the pilot members. The most important point is that generative AI risk-informed model credibility can be assessed, while methods and frameworks for credibility assessment of generative models are still converging. The work done here demonstrates a framework for performing credibility assessment that fits with and alongside existing accepted methods for physics-based simulations. And the second point is that model evaluation should be done in context and fit for purpose. We learned that the credibility assessment must be tied to a specific context of use to avoid overloading credibility of generative models. And synthetic data validation should reflect fit for purpose rather than a binary state. The third point is that risk-informed gradation of credibility factors helps transparency. And this allows developers to transparently communicate the maturity and trustworthiness of their generative models, guiding future users in determining what additional validation is needed for their own purposes. And by transparency, we mean that clarity about what the model does, how it was validated, and what users, such as regulators, clinicians, or AI developers can reliably trust it for. And the last point is that as all data-driven models, lifecycle maintenance is critical for generative models. As data, context of use, or clinical needs evolve, ongoing validation matched with the model risk is necessary to ensure generative models remain fit for the purpose. Yeah, that's done. And now we welcome your questions, this Q&A session.
So thanks, thanks a lot, Arezou. That's really helpful. So these pilots is different to the many others that we've been done so far, in the sense that we are looking not at the mechanistic model necessarily, but a more machine learning model. But I also know that there is a lot of colleagues who have expressed interest in this particular domain. I would like to ask all the various members of the panel, David and others, to come forward. And I think for everybody, you have the opportunity to either put questions through the chat that we can, I'm going to check with Lara that we have the operational Q&A functional, and try to explore a bit together questions. I'm going to throw a question myself just to kick the discussion, but hopefully give the opportunity to colleagues in the audience to process. It has been quite I mean, I know, because I follow this more or less closely, that it has been quite an intense period of weeks of discussions between many of you. And I know as well that there is a sense that we still need to do a bit more work to fully sort of unpick all the complexities. So this is more work in progress than a fully finalized sort of, like a task and finish type of group so far. But what could I ask, for David and Gavin and Daniel and Steve and others that are online, Mark, who was also quite engaged, and some of the other colleagues that were part of the group, which are the areas where you think there was the biggest difference to you compared to, between running the credibility assessment guidelines from FDA and the ASME BMB40 compared to do this in mechanistic models, which is for what it was originally conceived?
Maybe I'll pick that up, Alex, to start with. I think from a regulatory perspective, this was slightly unusual in that we had this in silico population, if you like, that I think for us, this was perhaps a step further than we'd considered. I think from a regulatory, we're used to looking, if you like, or we have experience of standard mechanistic models being used to, look at the way devices perform. And this one I think was really interesting because we were able to, certainly from my point of view, tease out maybe some of the weaknesses we see in normal clinical trials, that we were able to, the team here were able to kind of convince me that there was a way of looking at these patients that were perhaps underrepresented and complex things, perivalvular leak, all sorts of things that I, as a neurosurgeon, don't fully understand. But I think the message here was we were able to, provide this data in a way that regulators could look at it and say, well, look, it's transparent, it's been assessed, it's been validated, and ultimately the risk of using it has been quantified. And I think for perhaps us sitting, if you like, on the regulatory side, that was a really useful thing. As you say, rightly, there's more work to do. This is going to be supportive at the minute, but I definitely see this in the context of those underrepresented groups, those small orphan device scenarios. This is really something I think we can, use going forward. And I think it's on all of us to make sure that we don't, miss that opportunity. And I think, that's what I would say. And I suppose as a clinician as well, we want to get these difficult to treat and hard to reach patient groups to be front and center of our thinking and to innovate for them.
Thank you, Gavin. David, would you like to add something from the point of view? I mean, you are experiencing sort of modeling and simulation and in looking at how to apply these sort of standards for credibility assessment, but I reckon that that in a sense is a bit different to what we have done here, that the regular sort of application of BMB40. So is there a specific sort of lessons or specific observations you made that you think are worth sort of unpicking here?
I think that, as a medical device manufacturer, we're always trying to cover the patient population, right? So we might make 4 or 5 devices of various sizes and hope that, they could fit everybody that would need, say, in this case, a heart valve. So I think that one of the best ways to get at the answer to that question is with virtual patients, right? Where you can look at extremes, you can look at, what if I combine these variables? Does it create a different patient that, might not be covered by the sizes that I have right now? Usually the older way to do this was just to look at, what, competitors had for sizes, or to look at what, what you had for patient data scans from doctors, and then, try to, on a limited data set, guess how many, device sizes you need, again, to cover the patient population. So I think that this gives us so much more power in answering that question and providing the right amount of devices, for everybody.
And do you think so? There's a question in the chat by Marta Dacunia that is saying, would this sort of proposed model be applicable to ultra rare diseases? And when I saw the question, and by the way, I think the Zoom is set up in such a way that anybody can unmute and put your camera as well. So what I would suggest, after taking Marta and Marta's question, if anybody wants to ask, you can just raise your hand, and it will be a bit more interactive than doing via text, and probably more amenable for everyone. But am I right in Aresu that the answer to Marta's question about would this proposed model be applicable to ultra rare diseases has like 2 parts. One is the methodology you explained for how you would go validation and verification. I think my understanding is that the methodology will be equally applicable. The problem is that your model may not be valid in ultra rare conditions if your data set is not good enough. So what we have done here is validate the quality of the generative model. We haven't, which indirectly may end up telling us that our data set was insufficient. Is that your understanding as well, Aresu? Do you want to comment on that?
Yeah, exactly. This is exactly what I see. And for ultra rare diseases, we basically struggle to find adequate data to train the generative model, and there is a kind of severe bias in the training data. Then we need to deal with this with some strategies for sorting out this type of bias in the data, and then in the validation step again, we still have this challenge. So Yeah.
And Daniel, just following it up a bit with that Marta's question, and then Marta, I'm happy for you to chip in if you want to expand on that initial question. But I'm wondering, imagine now say that MHRA would sort of, as regulators, would advocate nationally for the data sets that are being collected, like our Future Health, the UK Biobank data sets, like the new National Health Data Service that is being developed, that those would be accessible for regulatory centers like the CEiRSI, not just ours, but also, let's say, the AI one. Would that be a way to potentially address part of the problem of rare or ultra rare conditions, in the sense that they're rare when you are thinking about relatively small sets, but if we would have full coverage of the country, that might be actually partly overcome by combining them. And is that part of what we should be doing, do you think?
I think that certainly seems reasonable. If the model shows that you don't have enough ground level data, then, and you'll have to forgive me, this is not my particular area, but I know the MHRA has places like CPRD, which is I believe it's the Clinical Research Practice Data Link, yeah. which is set up and designed specifically for sort of making data available for trials and for research in this sort of manner. If that's something that would progress being able to assist building these models, then I can certainly see that's something that we as the MHRA could be for supporting.
Super. I mean, I see Alexander as well from MHRA. Is anything you want to add to that? I mean, just be mindful that the CPRD is primary care data. So, for instance, it doesn't have access to things like imaging that were central into some of the things that AirSI show. So the notion of something like CPRD, in the sense that is national, that is large scale, as opposed of the specific modalities that today are present in CPRD, is perhaps what we are discussing, but therefore expanding to that, right?
Yeah, sorry, you'll have to forgive me. I'm not as close to CPRD as some of my other colleagues are. But certainly if there are other, let's say, data assets that could be leveraged in this way, then I'm sure within, the bounds of our legal sort of jurisdiction and boundaries, I'm sure that's something that we'd be able to support. I'm not sure if Alex did have anything else that he wanted to add.
Thanks, Daniel. Not on that particular point. It's not my area of expertise. Hello, everyone. I'm Alexander McLaren. I'm one of the benefit risk evaluation assessors in the safety and surveillance team, looking after cardiac implants and other types of cardiac devices. Really, I just have some general comments that the presentations have been great and it's been fascinating. And I wanted to agree with the comments that Gavin raised from his perspective. So from a regulatory perspective. So all of this really, I imagine, is a great complement to traditional studies that we've been used to, and perhaps reducing the need for early stage animal or human trials, of course, and prospect of modeling human anatomy and physiology to simulate device interaction and thereby giving or generating synthetic patient data in order to say, I don't know, stress test implants or wearables virtually is an exciting prospect. But I think we're obviously far from a situation where we could be, where this could be a full substitute for in vivo validation. However, I suspect as more time progresses and we gather more knowledge and experience, and more validated models are established, the more routine this acceptance of this becomes, and trust will follow. So as regulators, like the MHRA, will understandably remain cautious and vigilant on a number of critical areas to ensure safety, efficacy, and accountability, and regulatory compliance. So I imagine some of these critical areas, several of which have been addressed in these talks today, will include model transparency and explainability. So all these AI decisions, modeling decisions affecting design, testing need to be clear and explainable. And I think that's been raised in some of these talks. And there's also data and quality bias mitigation. So we need to ensure that data used to train these Gen AI models is diverse, representative, and clinically relevant, and that bias detection and mitigation strategies are in place. We also need to ensure that AI-generated designs or test simulations are clinically validated. Again, I think we went into detail here. So there should be requirements for external clinical validation of Gen AI outputs. Standards are in place for test scenario reproducibility and auditability. And I think finally, really, other critical areas are the traceability and audit trails in case of device failure or safety concerns. Traceability is critical. Also, cybersecurity and data protection, this may be a factor. I'm not sure, but we have to ensure that there's, of course, human oversight or human in the loop, where we require human validation for any safety critical decisions, and we define accountability between AI systems and human operator. So those are some of my thoughts. I'm by no means an expert in all of this. I'm playing catch up. So don't push me on any of those points, but those are some of the considerations I've thought about, because I'm used to looking at clinical trial applications. So this is very interesting and exciting. But of course, I'm relying on experts here with much greater knowledge in this area than me.
Thanks a lot, Alexander. And I'd like to well, it's great to see all of you, by the way, and great questions so far. So I'd like to move on to a couple of questions, which I think are around similar themes that Marta Suarez has put. So rather than me reading them aloud, maybe Marta, do you want to put it yourself? I think it's going to be more interesting just to hear it from yourself directly. Okay, now we're not hearing you. While I give the chance to Marta to sit up, I'm going to try to read out the question. So the first question is around sources of uncertainty have been mentioned and distinguish statistical from epistemic. Was the structural model uncertainty considered? And is this applicable to these non-mechanistic type of models? So
Shakun want to answer?
Say again? Yeah.
Shakun, do you want to answer, or can I say something?
Yeah, for the could you please repeat once again?
Yeah, I think Shokun, the question is aleatoric versus epistemic uncertainty, and I think most of the aspects that you explain are actually more on the statistical side, if I'm not mistaken. I think the epistemic component, which in non-mechanistic, purely data-driven, is about how we measure all the things that are important, right? Because ultimately you can have, we are pulling all the data, Marta, together in this generative model. So essentially we have everything and only what we have provided to it, right?
Yeah.
So is there any other consideration, Shakun, that you want to add?
So
yeah, go on.
Can you hear me now?
Yes, very good.
Oh, perfect. So if I can maybe add to that.
Yeah.
So I know that these methods do average over different model configurations. What I'm unsure is whether, because my impression, and I don't know much about this, is that that uncertainty actually isn't integrated as epistemic or statistical. So the uncertainty over the model averaging element. And I think only Bayesian methods of would allow for that structural uncertainty to be integrated. So I guess the question is whether there's maybe an element of uncertainty that isn't characterized.
So is it a question about characterization, or is it the question about whether one can build the models with that?
Yeah, exactly. In generative models, epistemic uncertainties, which are related to the structure of the model, can be considered. And as Shakun listed here, we can consider, for example, uncertainties in the parameters of the model, and this related directly to the structure of the network. So there are some techniques that can analytically measure these uncertainties in the parameters, like Bayesian frameworks. These are specifically can work on the parameters of the network. And then also about the structure of the network, the uncertainties in the design of the network, and even loss functions, hyperparameters that we use in the loss functions, all of these, and even the distributions we define for the latent space, all of these can be considered epistemic uncertainties that are related to the structure of the generative model and can be modeled and be assessed in this context.
And these are the things that we tend to measure with things like what we call ablation studies, where we look at sensitivity to the output. For instance, say that we use as a metric of quality some sort of a statistical test of overlap between the distributions, the target distribution and my empirical distribution, right? So you can start modifying all of those parameters that Arezzo was talking before, and look at how changes in the output of that metric are affected. So that will give you an idea whether, for instance, it matters so much, the particular decisions you did, or whether the results are fairly stable. And that tends to be fairly common, that we do that in good papers, let's say, of any deep learning work. Sort of the text will tell you that you need to do sensitivity so that you know that you're not just showing the result to a very particular arbitrary design of your network, right? So whether these are generative models for virtual populations or are generative models for any other work, we tend to do that as kind of default, right? I see that Gloria is with us, who is also expert in all of these areas. And I say, is there things that you want to add about this? I mean, you know as much as many of us on all of these techniques. So.
Thank you, Alex. Very, very interesting presentation. No, I was wondering, so the validation of these models, it's performed at the output. So with respect to normative studies of normal populations, let's say, of reference populations. So I was wondering whether there is anything going on in the latent space, because I saw the model, which is an autoencoder with many inputs and many output channels. So maybe if we focus on the latent space, we could distill some relevant information, or maybe have some more informative features. What do you think?
Yes, so just to give a bit more context, so this whole exercise was, we took one particular network where the goal was not so much to propose this network as the right one, but to show with a network like this or any similar one, the process you will go about to demonstrate the quality of the generative model from a virtual population point of view. So one aspect that I know Arezzo and colleagues spend quite a bit of time in the preparatory meetings is to say, should we use also metrics about the outcome of the trial, or should we use metrics about the simulations that you do with each of these populations? And the conclusion was that we shouldn't do that, because you're otherwise conflating multiple things. Like the same way that when you do a human trial, you decide on your target population and you don't mix that with sort of the quality of the sensors you use to measure the outcomes. These are kind of different parts of your design that you need to be consistent, they're separate. So what we decided is that we wanted to make sure that the metric of the generative AI was purely related to the thing that the generative model is trying to represent, which is a target population. They don't need to be healthy subjects. It could be the population of your disease patients that you're going to be treating with these valves, right? And from that point of view, I just gave an example about the overlaps of distributions, but it could be any other test of significance between multi-dimensional distribution similarity tests, so to say. Now, I think what you are suggesting, if I understood correctly, is to look at these joint latent spaces more from a point of view of interpretability and from explicability, right? So are we generating features that have some insights about those phenotypes? And that indeed is potentially interesting. I'm not sure from a regulatory point of view if that would be relevant, but that's more perhaps for Daniel and Alex and others to I mean, when you receive a trial, Alexander, to analyze, how do you assess the quality of the cohort that they have chosen for a human trial? I mean, is it based on previous evidence of where that device would be relevant, or how do you know that they haven't constrained it too much or relaxed it too much?
So we're talking about the intended cohort of a human device.
Trial. Yeah, for a human trial, whatever device, say a device X, we can carry on with the example of the TAVI here, just for simplicity. Someone comes with a trial and says, look, we want to test this new valve, and comes with a population that you say, this is too constrained. So the inclusion and exclusion criteria are so narrow that this won't be any representation of what's going to happen in the general public, or the other way around. They're so lax that.
Yeah, I mean, it varies. And of course, if we're talking about just a standard TAVI device, then we would expect not to be any issues with the population or gathering enough subjects to undertake the trial. But sometimes there are disease conditions where there's a much smaller population, so we might not expect the manufacturer to get the full 400 patient years as is required in the TAVI standard. You accumulate that much data, but I mean, it really does depend on each on its own merits, case by case basis, really. And then, of course, you have to look at the number of centres being. Obviously, we just look at the UK, and so we look at the number of centres being utilised, employed, because it's important that there's obviously a learning curve for clinicians, and that we can't have too few patients in each centre. There's a fine line between not enough patients per centre, having too many centres. Obviously, advantageous for the manufacturer to have a lot of centres, because it means that they can accumulate those subjects in a much quicker period of time. But it does result in a prospect of perhaps not enough patients being treated per centre. So there's not that good learning curve for the clinicians in the individual centres. They don't get that experience. But yeah, I think it's a case by case basis and depends on the device and on the condition being treated. But inevitably, there's a lot of similarities in these patient cohorts, and I don't know if it's more of an art than a science, I suppose, personally speaking, because of my experience, and I don't know if this is written down. It's a good point. It might be written down in a pro forma somewhere, but it's a tricky one to answer.
I think what you're saying is that perhaps there's a lot of considerations into the product that you're going to be testing on that population, and practicalities about, can I acquire sufficient cases if I become too strict? If I need to do it multi-center, will I have enough cases per size for them to develop experience so that there is no confounder related to the lack of, the learning curve might be still too early on. So that's what I would suspect, Gloria, that a lot of the discussions about the validation of the cohort are more related to reproducibility in statistical terms of the target group and practical aspects, rather than sort of interpretability in terms of the phenotype, which is what we will find in the probably in the latent space, right?
Yeah, in the sense that in the latent space, you can also build the statistical distribution of the reference population, no? As we do, for instance, when you compare healthy subjects with diseased populations. So instead of doing this in the output or input space, you can do it in the latent space in a kind of feature extraction previous step. This was what I was meaning, yeah.
That's a great point. And I think to some extent, this is something that my suspicion is that is not done currently in human trials, because you don't have the tools to do it. I think what you're saying is that if you were to group patients into subgroups for some sort of a stratification of the therapy, you might be able to do that in the joint latent space, as opposed to doing in the space of whatever data you collected. But in general, unless they're going through a generative model like this, they don't have access to the latent space. So normally, I don't think they're doing it at the minute. But that's a great point, actually.
Yeah. Thank you.
Super. I don't know, Marta, if we cover the second part of your question as well. Do you want to formulate that a bit?
Yes. Can you hear me now?
Yeah.
Yes. Okay, perfect. So I was just thinking about the potential for in silico evidence to be used in smaller population groups or unrepresented population groups, which typically are smaller patient groups. And we were discussing earlier how to source evidence for those groups. So I guess the question is, how do we first establish credibility? But if there is uncertainty over credibility of the model, how should regulators respond to that uncertainty? Whether they should postpone approval or request further data collection. So it's just trying to reflect on the inherent problem here, where you have small populations with which to develop your model. That means there may be uncertainty over credibility, and there may be uncertainty over the value of adoption. So I was just trying to get the regulation representatives here to maybe give some thoughts about this kind of inherent evidence issue.
Alex, maybe, or Alexander, both Alex, maybe that one's really a notified body question, because I guess we see the evidence in its totality. We don't approve clinical trials. That's up to the competent authorities. So what we're faced with often is a totality of evidence that's been collected for a device for approval. And then I think what you're talking about, Marta, is extensions to scope. So can this device then be used in a pediatric population or in a specific group that maybe wasn't part of the original, and how do we support that? I think for us, it's always a question of being pragmatic and being realistic. All the notified bodies are required to have clinicians. The large ones typically have experienced clinicians, and that may just mean old, but typically, they are experienced. We recognize the limitations, and as you and Alex have touched on before, it may be that you simply have no source data. There's very limited imaging data available in a small group. We can be reasonably pragmatic and say, look, provided the evidence is transparent, we're aware that there are limitations, we can issue certificates with conditions that would then require the manufacturers to report back, what's been happening to their device as the data is gathered. So there are ways that we can be pragmatic. I think I know what you mean, and I know what you're getting at. There is a degree of variability inevitably with notified and approved bodies, because they're independent organizations, and it is something I think that Team NB and Team AB are are trying to address. And I think some of the guidance, particularly from, the MHRA and the Commission recently, has been to say, look, try and approach this in a pragmatic way, look at the weaknesses, assess what they are, and do you think, that the patients ultimately are going to benefit from it? And I suppose for us, if it's a rare condition with very limited treatment options, even if the risk is potentially higher of complications, is that better than no treatment? And I suppose that's what we do as clinicians all the time. We weigh that benefit risk for the individual patient. And I think sometimes we do have to be a bit pragmatic. I think you're right, that the evidence may not be perfect, but provided there's a realistic and a transparent process ongoing to try and collect that evidence to support it, then, I think we should be pragmatic.
And that this is probably applying to both human trials, when you have a trial that then you may need to do a subsequent phase of the human trial, or the situation where you have part of the evidence is in silico, but then you look for sort of post-marketing evidence as a confirmatory one, or potentially to go and do additional data, evidence gathering, right? So it's more about this totality of evidence that you were bringing, rather than whether the evidence is obtained with one modality or another one only, right? Which is a great point to bring John, who came online now, came on with the camera now, because that's exactly what he's thinking in his work. So perhaps, John, if you could briefly introduce yourself, and then see how all of the sort of things that we've been discussing in terms of how to operationalize estimating the quality of the evidence we have, or the quality of our models, could be put into that sort of more holistic framework that you're working on.
Yes, thanks, Alex. So I'm John Williamson in the philosophy department at the University of Manchester. I'm interested in ways to integrate different evidence streams, particularly when it comes to the sort of regulatory aspect. So just to give a bit of background, one area that I've looked at quite a lot in the past has been carcinogenicity assessment. In carcinogenicity assessment, organizations such as the International Agency for Research on Cancer, they routinely integrate mechanistic evidence together with animal studies and human studies when trying to decide whether a chemical is carcinogenic, for example. So it's a kind of well-established procedure there. And what I'm interested in particular, is whether some of the lessons that they've learned over the years can be applied to the setting of in silico trials, because in silico trials can often be viewed as mechanistic studies, and then the question arises as how you integrate those mechanistic studies together with human clinical studies when you're trying to make regulatory decisions. And so a lot of work has been done on this in other areas, and I'm interested to see kind of the extent to which this might cross over. So I'm finding this conversation particularly fascinating. And I'm interested in aspects like how you assess the reliability of these in silico trials. So I was interested in the scorecard approach, for example, the 7 Cs that we heard about earlier. And I think this is all essential to having a proper method of integrating in silico studies with clinical studies when you're coming to regulatory decision making.
Thank you. And I think it's important, the work that yourself and other colleagues are doing in health, but also in other non-health domains, because when you start seeing that there is a consistency in the approach across different domains, that hints you that there are fundamental principles behind that, and it's not just an artifact of the specific use case you're looking at, right? And that's partly what we're trying to do with this sequence of case studies that we are exploring, these various projects. Stephen, I'm wondering whether you want to add some comments. You've been very active in the contributions to the event.
No, yeah, I appreciate that. Yeah, I think, what's really interesting about all this is, to me at least, and I think this is a little bit afield from what was just discussed. Obviously, the quality of the information relative to clinical trials, I do think that gets to some of what Renee was talking about and describing in his contribution. I completely echo the value of the insights gained, the mechanistic aspects of having the suite of information that you have by nature of doing an in silico clinical trial. You're able to define so many features and measure so many things with respect to individual virtual patients. So I think all that's really important and valuable, and certainly echo the comments there. To me, on this pilot, probably the biggest thing that got me thinking, and I think was a topic of quite a bit of conversation with respect to the implementation of these methods and how regulators might interpret the information and need to consider the information, is really, and I think somewhat gets to the traceability, but it's this concept of these generative models being a portion of a pipeline for an in silico clinical trial. And the fact that, fundamentally what we're doing here with respect to this model as described in this pilot, and the extensions of it as you think about other aspects of input data, is, you're essentially generating that input information for downstream simulations. And I think there's a lot of interesting applications and considerations there. We talked about the tunability of the virtual patient cohort and how these methods provide the ability essentially to tune into different populations and so forth, assuming you have, appropriate training data and so forth. But I think the value and really the questions and the challenges associated with how do you package the credibility information for a generative approach like this, and be able to pass it down to subsequent activities in a pipeline, is really interesting. And I think we talked a bit about this notion of considering the credibility assessment for this generative model in isolation. And I think the value, and Andrew or Alex, sorry, you mentioned that we've landed on this idea that you want to be able to assess the credibility for the generative model in isolation. I'm a proponent of that. I agree with that. I think there's a lot of value, particularly because you could put some of these models on the shelf, if you will, and pull them off as necessary in other applications and so forth. So I think there's important aspects to assessing credibility in isolation. But what this really speaks to me about is, how do you document that? How do you really establish the credibility bounds, if you will, of the generative model and what was produced and what the applicability is? There's a bit of discussion with task-specific validation. There's some notes there in terms of how you might think about the credibility assessment for the generative models, in terms of what their application is going to be. And I think that's an area that, as a community, we really need to flesh out and think carefully about, in terms of how do you, how do you recognize the value of these approaches being package deals, in a sense, of being able to create these virtual patient cohorts and leverage them for multiple studies? I think there's industrial applications and benefits to doing that. I think there's regulatory applications. I think there's a lot of benefit to being able to say, yes, this virtual patient cohort is good, and you can use it in multiple studies. I think everybody would recognize the importance of that. But I think just logistically and practically speaking, how do we document that? How do we establish that? How do we put bounds on ways in which we can use these generatively created virtual patient cohorts in subsequent analyses? And what are some of the challenges associated with that? Because I think the potential for these methods is enormous. A lot of the technical details fly way over my head. But I think in thinking about how one would use these, it's clear that these methods are fantastic and provide great opportunity for at the very least for the physics-based simulation folks in using these. So I think grappling with these issues is going to be really important, critical, but also really valuable for what we do with these and what we're able to do with these.
Thank you, Stephen. Perhaps something that is sort of an idea that is forming as I see hear you speaking about this is that the fact that we want to disentangle the downstream use from the generative model itself doesn't mean that maybe there is more space to provide additional metrics of the generative model that helps us to understand the level of uncertainty, so you can do sort of uncertainty propagation through a pipeline if need be, right? I mean, I think you have similar uncertainties in any trial. It's just that we perhaps are not so acutely aware, right? So, for instance, we say, okay, we're going to measure some endpoint from our trial. That as we go more towards a world where we use sort of EPROMs and all of these different ways of measuring patient-reported outcomes that are based on wearables, you know, we could have different quality of measurement systems in each of the wearable technologies, and that will have an impact as well on everything else you're doing. And we might need to start specifying those a lot more than we're doing at the moment when we design a trial. So the fact that we have concatenation of sources of error is already a fact. It's just that perhaps, you know, some of these techniques are putting that more to the front, right?
Yeah, absolutely. And, you know, I know you like thinking about systems of models and how different things come together. And, you know, I think that's absolutely true in this case. And I think this is a very practical example of just even two models being linked together in series here, in the sense that you're, you know, fundamentally generating the virtual patient cohort and then you're running some simulations off of it. You know, there's all of these other things you point out, the digital health data, there's all of these other aspects of different types of information, the quality of that information, and really trying to grapple with and be able to quantify and be able to understand the trade-offs of different evidence generation approaches, and trying to put these things in conversation with each other and considering them at the same time and considering the pros and cons of each method. And having kind of a standardized way to assess, you know, in some level, evidence credibility across the different types of evidence that you're going to get. You know, we have to grapple with this in current applications as we're talking about here, but I think it's relevant to be thinking about this from a quantitative as well as a qualitative perspective with respect to more traditional sources of information, whether it's digital health data, you know, other types of information, or more traditional clinical evidence.
Excellent. So we're coming towards the end of our session. Maybe just one last question open to whoever wants to help answering that. So as you were talking, Steve, I was thinking a trial, a human trial, or is essentially an experiment, a control experiment, to assess what ultimately is a very complex system, because you have interactions between multiple moving parts in that. You know, the patients, the devices, the clinicians being involved, the measurement systems from the outcome. So there is a lot of things there. And In-Silico trial is also a complex system because there is many models and submodels that are orchestrated in some sort of pipeline, right? To what extent, you know, there is a difference between these two, or is just that In-Silico technologies force you to be more explicit about that complexity, as opposed of a statistical design of a randomized control trials, which somehow hides everything under, you know, some statistical numbers. And this is a bit of a question as well for you, Marta. Well, it's nice to see that you have your hand up, Marta, because I wanted to ask you this, you know, don't you think there is also a lot of fundamental epistemic uncertainties that we're not necessarily incorporating in a human trial, you know, beyond the obvious ones that we normally try to capture, but, you know, regarding measurements, regarding, you know, other aspects as well there, Marta?
Sorry, just to try and be very quick replying to your question. I think the key difference, the key element here is causality. In a clinical trial, you're trying to compare and attribute causality to the effects of a particular technology in relation to another. Attribution of causality in a randomized clinical trial, comparative clinical trial, is, as you suggest, statistical, but it's a perfect means. It's via randomization. Now, obviously, for policy, that randomization may have limitations because the patient population may not be representative, but you do have a causal effect if your design and your protocol is generalizable and representative. You may not be able to explain the causal relationship, which is why in silico mechanistic models might actually add value, because if you could understand the causality mechanism, you could extrapolate to other groups, other populations, et cetera, et cetera, et cetera. With a clinical trial, you have that design causality, but you can't explain it. With the mechanistic model, you have a hypothetical causal mechanism, and there's uncertainty, and that's why I was kind of raising the question about structural uncertainty. Is actually what we're caring about is that causal mechanism, is how the treatment can lead to different outcomes in relation to another treatment, and that causal mechanism is key. And in an in silico mechanistic model, there might be structural uncertainty about that mechanism. So structural uncertainty does matter, maybe perhaps more, because of the impact on causality. Sorry, I don't know if that contributed to your answers, but those are my reflections on the key differences between a mechanistic and a statistical or a design-oriented causal effect.
Thank you. Any other final thought about that question before we move to the closing sort of? Okay, I don't see other hands up. So just so Aresu asked me to close with some next steps. And just to, well, first of all, I really like to appreciate, to say big thanks to quite a lot of people. I'm not gonna embarrass them by listing them by name. You saw the list at the beginning of the slides that Aresu had, but there is one person I can just pass without specifically thanking, who is Aresu, for having driven the whole team forward and, you know, sort of taking the time that was required to collect the ideas, put them together. We do, and what I'm saying we is also Aresu and myself, we both realized there is still a lot of digestion that we need to do. We had to put a date to this event. But what we want to do is we're going to continue with, so Aresu will follow up with the people who have been involved in the pilot with the intention of having a few additional sessions to try to bring the current document, include the current material that you've seen together with the discussions of today, into a possible sort of publications that explains a bit the process in a bit more detail. There are I's to be, you know, dotted and sort of T's to be crossed still. If there is additional people who have a particular interest to be involved, or if there is any of the colleagues who were involved in the pilot so far that for, you know, whatever reasons they don't have time or interest to continue in that subsequent phase, let Aresu and myself know. But otherwise, we will ask you once more to those involved. But if anybody who hasn't been involved wants to be more closely linked, just let us know. We will, as I said, try to put this together into a publication like we're doing with all of the pilots. And then in the horizon, and this is more an aspiration at the moment than something that we are necessarily going to do in the CEiRSI, will depend on possible funding as well for the next phase. But we would like to take one of these, sort of from the 10 pilots we're doing, about, you know, perhaps 3 of them into an implementation phase. And this one seems to be quite an important one as well. So we will come back to all of you. So with that, I'd like to also remind, Lara requested me to remind you all that there will be no further seminar in July, as far as we know. We are trying to close an additional one of a radiotherapy pilot in August potentially, but otherwise most of the activity will continue in September. We had yesterday a really interesting webinar looking into the epistemics and the ethics of modeling and simulation in this area that I recommend you to watch once it comes online. And there is going to be some additional webinars that we are designing for Q4 If you have suggestions or ideas or want to be involved in any of them or help develop one of them, by all means, let us know, because we want this to be a community-driven endeavor. Okay. So with that, thanks to everyone. Have a great rest of the day, and thanks a lot for your participation.